Hello, everyone. Good afternoon. Thank you for coming for this lecture. I'm really glad to have a Professor Nicolai Lehnert from University of Michigan here. He's a guy that I know now more than 10 years, I guess, like 2014. I went to a conference and met him. So since then, we exchanging emails, and also we had a student come in. It's from Professor Daniel's lab, and he was a sandwich student there for 6 months, I guess. So it's a pleasure to have him here. He's a guy that works with a lot of cool stuff, a lot of amazing spectroscopies, actually, mainly on iron. And I'll talk a little bit about him. So he finished his grad in chemistry at Düsseldorf University, and then he did a PhD with Professor Felix Tuxek and Felix Göttlich lab in Mainz University. That's it. Working on some nitrogenase, like 1999. Then he went for a postdoc in Stanford for 2 years with Professor Ed Solomon, doing basically a lot of MCD and DFT stuff. So in 2001, he went for a position as a senior researcher at Kiel University in Germany. And then in 2006, he came back to US and is there since then as a professor in University of Michigan. So his main focus is on bioinorganic chemistry using like a model complex and enzymes, mainly related to nitric oxide and also the nitrogen cycle. I'd like to thank Caterina that's here. So he is in Brazil because of the summer school of Federal University, and Caterina made the first contact with him. So we got the opportunity to have him here at University of São Paulo. So thanks, Caterina. He's gonna give a talk, actually 5 days, a course in the afternoons at Federal University. I'm not sure if you're able to attend right now. Should have like applied for this before, but he's going to be there and kind of have some time, maybe here in the mornings to discuss with people. Not sure about the time, but so thank you. It's a pleasure.
Thank you. Okay. This is very loud. Maybe I can try to move this away. Is that better? All right. Yeah, thank you for the nice introduction. Thank you, Caterina, for bringing me out to Brazil, my first time in Brazil, so that is exciting. And Daniel and Antonio for hosting me here. So today I will tell you about our work on nitric oxide activation by diiron complexes, which is relevant for the chemistry of these flavodiiron nitric oxide reductases. So here is, oh, wrong side. Here's a little overview of my presentation. So first of all, I will tell you about these flavodiiron nitric oxide reductases that we are interested in, and we want to understand, their mechanism. And then I'll show you some previous work we've done on model complexes where we identified this semi-reduced pathway as a possible mechanism. And then in more newer work now, we investigated how the exact structure of this diiron dinitrosyl core actually affects reactivity, right? So what is the exact structure? And we distorted that core and we found a new reaction that is a non-biological reaction, making these dinitrosyl ion complexes. And then I will talk a little bit about what we think the mechanism is of how these dinitrosyl ion complexes in general form when you reduce these iron nitrosyls. And then I have a little bonus chapter, so depending on how we're doing, if we have some time, I can talk about iron hyponitride chemistry, or I can skip if time is up, right? And then I'll conclude my presentation. Yeah, so as Antonio was saying, my area of research is bioinorganic chemistry, and essentially we have 3 cornerstones in this field. Of course, we're interested in metalloenzymes, so there is the enzymology in biochemistry, but an alternative way to look at mechanism is using synthetic biomimetic model complexes that we can make, and they mimic the active site of the enzyme, so we can study the chemistry that the enzyme does by using an inorganic model system. and we have more control over the properties, the structural properties, the electronic properties of these model complexes, so we can essentially study this chemistry in kind of a broader way than what you see in the enzyme. And in order to study reactions, you need to kind of look, into your flask and see what's going on. So you need spectroscopy, and then we can supplement this also with quantum chemistry. So these are kind of the cornerstones of the field of bioinorganic chemistry. That's why it's so much fun to be in there, because there's all these different things you can do. Okay, so the molecule we're interested in is nitric oxide. If you look up nitric oxide on Wikipedia or something, nitric oxide is a very toxic molecule. It's poisonous. It's produced in fossil fuel combustion, right? It's part of smog. So it's a molecule that has kind of a bad reputation. But that changed then back in the 1980s when it was discovered that nitric oxide is actually made in our bodies, and it has very important function in signaling, for example, right? And here's one example. So the so-called endothelial cells here in blue, these are the cells that line the inside of your arteries. They produce nitric oxide. Nitric oxide is released in the smooth muscle tissue. It relaxes the smooth muscle tissue and ultimately leads to a dilation of your artery, right? So if you have a low amount of nitric oxide, your artery is constrained. If you have a larger amount, it dilates, and that means it lowers your blood pressure. So nitric oxide controls the blood pressure in the human body, in the arteries, through this mechanism. You also make nitric oxide in your brain, for all places, despite the fact it's so toxic, and it's involved in signal transduction. So when neurons communicate with each other, nitric oxide is one of the signaling molecules. And the way that our body handles the toxicity is by using these very low nanomolar concentrations. And then ironically, on top of this, our body actually makes use of the fact that nitric oxide is toxic and uses it for immune defense. So your immune cells, for example, the macrophage here, can now make higher concentrations of NO, micromolar concentrations, to kill invading pathogens. So it's a very important anti-infectory agent that our body produces, an important part of immune defense. And taking advantage of the fact that it's toxic, and you can also use it, right, for cancer therapy, trying to kill cells with NO-releasing molecules and things like this. But this is not what I'm going to talk about today. We're going to stay with the aspect here of immune defense. So we can say nitric oxide is a very potent immune defense agent. But very interestingly, just like with man-made antibiotics, right, antibiotics that we make like penicillin, some pathogens over time evolve defense mechanisms against them, and they become immune to our antibiotics. And the same happens with nitric oxide. So in the case of nitric oxide, there are some pathogens that produce these flavodiiron proteins as protecting them from NO toxicity. And that was first discovered by Gardner in 2002. So he studied some E. coli strains, and then he would take these bacterial cultures, he exposes them to NO gas, and then he sees that there's a transcription factor that they have that senses NO, and once NO is detected, they make these flavodiiron proteins to defend themselves from NO toxicity, right? So they can essentially fight off our immune response, they can survive, and they can lead to infections that are more difficult to cure for our immune system. So these flavodiiron proteins are known for some time. They're known to be oxygen reducers in microanaerobes. And so the idea here is that some pathogenic bacteria have essentially adopted those enzymes through evolution to now catalyze a new reaction, which is NO reduction. They take 2 NO, 2 electrons, 2 protons, and they make nitrous oxide and water. And nitrous oxide is not very toxic to pathogenic bacteria, right? So they can survive and they can proliferate in our body and lead to infections that are harder to cure. So that's kind of what got us interested in that, kind of this reaction here, and thinking about how that reaction works for these particular enzymes. So for all bioinorganic studies, the first starting point is to take a look at the catalyst, right? To look at the reaction center or the active site of the enzyme. And here is a crystal structure of a flavodiiron NO reductase. You can see here we have this non-heme diiron core in the active site. So we have 2 iron centers, and they are coordinated by amino acid side chains. So 2 histidines, here's an aspartate, a glutamate, and then there's a bridging aspartate. This bridging aspartate will turn out to be extremely important for the mechanism, and we'll get to that. And then if you kind of look at the structure for a while, you see that both ion centers are only 5 coordinate. So there's 2 open coordination sites, and they're right next to each other. So as a bioinorganic chemist, you can start thinking about the mechanism now, and you think, oh, I need 2 NOs, and I have 2 ions. Maybe I bind one on each ion, and they're right next to each other. I can couple them, maybe I can make N2O, right? So you can, just looking at the structure as a bioinorganic chemist already gives you some mechanistic ideas. And then, go back here. The other component in the active site, this is a flavin cofactor. The flavin cofactor is an important cofactor in biology, and it's a redox active cofactor that can deliver electrons, 1 or 2 electrons, for a chemical process. So there is also this reductant here present in the active site. Okay, so in terms of the mechanism, we know that we start from the diferrous form of the active site. Both ions are reduced. And then in the first step of the reaction, 2 NOs come in. And that was determined by some very great work by Don Kurtz and his group, published in 2014. And they did rapid freeze-quench Mössbauer spectroscopy. So they were trying to freeze intermediates on the reaction pathway. And they can see this intermediate form. And then the next step, they form N2O. So this is what we would call the catalytically competent intermediate, right? That is the intermediate that's responsible for the reaction. And that was reported the first time in this paper here. Now, from at this point, I want to take a little sidestep. You can see that I wrote this here in kind of a weird way. I wrote iron-free NO-. I have this FeNO7 nomenclature here, so I want to explain this a little bit. So take a little sidestep here, talk about these types of complexes. So NO, nitric oxide, has actually been studied in coordination chemistry for a very long time, and that's because nitric oxide is what we call a non-innocent ligand or redox active ligand. So that means when nitric oxide binds to a metal center, it can change oxidation state. It could be NO+, NO dot, or NO-, and a priori we do not know that. In this case here, where we have what we call non-heme ion centers and we bind NO, we know that these complexes are actually best described as an iron-free, that is high spin, with a bound triplet NO- ligand. And that was first determined way back in the 1990s by my postdoctoral mentor and his group at Stanford University. They did the first studies that showed that these complexes have this type of electron configuration, right? And so that highlights, of course, the idea that nitric oxide is a non-innocent. So when nitric oxide binds to iron 2, it oxidizes it to iron-free and becomes NO-. So we can also illustrate this electronic structure with this scheme. And if you have mononuclear complexes, you can study this very easy by EPR spectroscopy. So in this case, you can see how I wrote this. The spins are antiferromagnetically coupled. So we have S equals 5F, iron-free. We have a triplet NO-, antiferromagnetically coupled spins. Gives you a total spin of 3/2. You can take an EPR, and you can observe this directly for mononuclear complexes. Of course, in diiron complexes, the spins are coupled. So you can study it directly, but here you can. And I also want to introduce then this NMR-Feltham notation. So because NO is a non-innocent ligand, the electronic structure is always complicated. And so NMR-Feltham had the idea that we could just take the ion and the NO as one unit, right? We consider ion NO one covalent unit, and we count the valence electrons of that unit. So if we take an ion 2, we have 60 electrons. NO has 1 pi star electron. So if you just add this up, you have now 7 valence electrons, and so we call this FeNO7. So now we can call this an FeNO7 no matter what the exact electronic structure is, and now we can keep track of redox states. So if you oxidize FeNO7, you go to FeNO6. If we reduce, you go to FeNO8, right? We can keep track of the redox state in that way. Okay, so I think now we go, and then one aspect we were very interested in was the nature of this interaction, and I want to dwell on this too long, but we essentially studied a bunch of these mononuclear complexes, and we found that this NO- ligand is mostly a strong pi donor ligand. So it donates electron density back to the high-spin iron-free center, right? And you can see this by DFT calculation. If you calculate the molecular orbitals, these are the contour plots for these pi star orbitals of the NO- ligand. You can see there is a very significant amount of ion character mixed into these orbitals. The DFT calculation says it's about 35%, but because that number is not accurate, it's just a theoretical calculation. But what it shows is that the bond is very covalent, And that means these complexes tend to be very stable. That's interesting to note, because if we now go back to this mechanism, right? A number of scientists have prepared diiron dinitrosyl complexes like this, but what they found is that they're usually stable, so they actually don't really react to make N2O. In the original paper by Kurt's group, they proposed that this species is catalytically active. It can couple DNOs, make N2O, you go to the diferric, and then you can reduce this and circle back around catalytically. But these complexes tend to be stable, so an alternative mechanism is to involve the flavin in the reaction, right? The flavin cofactor is redox active, and so the flavin could provide one electron for the reaction, which we call semi-reduced. So then you go from FeNO7, FeNO7 to FeNO7, FeNO8 intermediate, one electron reduced. Or you could take 2 electrons from the flavin, then you get an FeNO8, FeNO8 intermediate. We call that super reduced. Right? So now the flavin plays a role in the reaction, and then from here you can circle back around and you do your catalysis. Right? If you look at this picture here, this is kind of what I call a reactivity landscape. Right? We're interested in now using model complexes to generally explore this type of chemistry. Right? Is there a way, for example, when we're here to direct going either this way or going either that way, and how does that depend on the electronic structure or the geometric structure of this species? How does it maybe depend on second coordination sphere effects, right? That could play a role here. And we can ask the question, are there other reactions you could do in this kind of reactivity landscape that nature doesn't do? So are there other non-biological reactions that could happen within that space, right? And that's what we're exploring in my lab. Okay, so if we do model chemistry, now what we're going to do is, here is again the picture of the active site of the enzyme, but we're going to use a synthetic complex. So we're going to make a ligand using organic chemistry. We're going to assemble a ligand that kind of reflects some of the key aspects of the active site, right? So for example, instead of the histidines, we take pyridines here and here. We have this backbone amine as a weaker ligand, replacing this carboxylate, and then very importantly, we have a bridging phenoxide instead of hydroxide, and we can install this carboxylate bridge, and then we have a model that kind of reflects the active site pretty well electronically. Then we know we need the diferrous complex. We can react this with FNO. We can look for this dinitrosyl compound, or we can look for, is there any reactivity that happens? But just like what I mentioned with what other scientists had observed, in our case as well, this complex forms, it's stable, and we can get a crystal structure. And the crystal structure looks like this. So here are the ions, here and here. Here is the ion NO unit, one of them, here is the other one. And you can see that they are coplanar, right? So the ion NO units, they're right next to each other. They're looking at each other, but they don't do anything, right? The compound is stable. They don't do the reaction. But what we were able to do with this complex here is we were able to incorporate this idea that DNOs should bind next to each other for that reaction to occur. Okay, and then we went on and looked at different chemical triggers, and this is all old work, so I don't want to talk about this too long. So just the most important result is if you take this complex and reduce it by 1 or 2 equivalents of reductant, you make N2O. So you can see here, this is a scale of N2O yield versus time. And this is determined by IR spectroscopy through gas headspace analysis. And you can see we add reductant, and after 60 seconds, we get 100% N2O. Right, so this complex is able to do that reaction once you figure out how to start that reaction. And the key here is to reduce the dinitrosyl complex, then the reaction will go. It doesn't matter whether you use 1 or 2 equivalents of reductant. You can see these curves are essentially the same. And so ultimately, it turns out you only need 1 equivalent of reductant. So this is an example for this semi-reduced pathway that I mentioned before. Once you figure that out, you can do that reaction now in all kinds of cool ways. So here's one that I really like, is spectroelectrochemistry. So you take a thin layer cell, right, with a mesh electrode, you stick it into your IR spectrometer, you hook it up to a potentiostat, you apply a potential, and now you can reduce the sample and take IR spectra at the same time. And then when you do this, you can see here, this is the NO stretching band of our precursor complex here. Then you reduce, this band goes away, and here is N2O being made at the same time. So the reaction actually is pretty fast and efficient. And then we also note for later, nothing else forms. So this is a very clean reaction. Only product is N2O. Okay, and so this complex then is the first example for the semi-reduced mechanism where we need one equivalent of reductant, one electron, to kick off that reaction, and that was published back in 2018. All right, so here is what's new. This is again the original compound, the same complex I just showed you, and I told you that the two iron NO units are coplanar. We can also measure this now. We can quantify that. For example, we can measure an angle. We go from nitrogen to iron to iron to nitrogen, and that angle is about 6 degrees, right? Again, reflecting the fact that the NOs are essentially coplanar. And then this complex makes, as you saw, 90% N2O in 1 minute. So my student, Amy Spielman, she had the idea, she asked the question, what happens if you don't add that bridging carboxylate here, right? So we metallate the ligand and then we add one equivalent of sodium acetate or propionate to install that bridge, but you don't have to do that. So you can take the ligand, you metallate with iron triflate and then add an O and crystallize it. You get this structure here. You can see there are two triflates now bound to the iron centers. And you notice one other thing, that one of the NOs here, this NO kind of comes out of the board, it comes out towards us, but the other one just goes straight down. So if you don't have the bridging carboxylate, the core will distort, it will rotate. So these two NOs, they don't like each other actually. You know, they're like two relatives you have, they don't get along. So when you invite them for dinner, you put one on one end of the table and the other one on the other end of the table, so they don't fight all the time, right? So if you give the option, these NOs will actually go away from each other. They don't like each other, and we can quantify that by using the same dihedral angle, which now is about 50 degrees. So they go pretty quite away from each other. And then when you reduce this complex now, you get some N2O, but then you also get some other product now. So you've now observed a new reaction that's not observed in biology by essentially distorting this core. That's all we did. Right? So once we discovered this reaction, we decided to investigate this a bit more broadly. So we made 3 different compounds with 3 different monodentate ligands. So the original one with triflate, we made one, this is 1-methylamidazole. Oh yeah, it's defined here. And then we made one with methanol as that ligand X here and here. And these 3 complexes are distinct by their NO stretching frequencies. If you take an IR spectrum, you can see here in this range that they all have different NO stretching frequencies. And the one compound that electronically matches the original one the best is the one with 1-methylamidazole. You can see with 1-methylamidazole, the NO stretch is 1756, right? Whereas, oh, wrong. Go back. This one's about 1760 something. So they are very similar. That means that electronically, the iron NO units are essentially the same, but the structure is different. So in order, if you want to compare reactivity, either coplanar or distorted, those are the best compounds to look at. In the paper, we investigated all, but I was going to focus on this for now. So again, IR spectroscopy tells you the whole story. So now what we can do is we can reduce the complex, and this is data for the complex of 1-methylamidazole. We reduce this with cobaltocene again, and we can observe by IR what happens. So the starting complex in solution now has this very broad NO stretching band, like this. And then you add one equivalent of reductant, one equivalent of cobaltocene. You get some N2O, and then you get 2 new signals. And then when you add another equivalent of reductant, those 2 signals will shift here and here. So from 1788 and 1715 to 1689 and 1632. Or you can just go and add 2 equivalents of reductant right off the bat, then you go straight to those signals here. And again, a little bit of N2O is made. Now, if you study iron NO chemistry, then these signals, you know what they are. These are so-called dinitrosyl iron complexes, where now you have one iron with 2 NOs bound to one iron center. And those can exist in 2 redox states. One is called the FeNO2 and the other one is called the FeNO2. And these are NO stretching frequencies for these complexes because they have 2 bands now because they're 2 NOs bound, right, to one iron center. And it turns out no matter what complex you use, the one with triflate or 1-methylmidazole or methanol, they all make exactly the same dinitrosyl iron complex. So that means that whatever monodentate ligand you started with is not bound to the ion that has the 2 NOs bound, because the frequencies are exactly the same. So that was one interesting result. So we've now opened up a new channel where, instead of the nitrogens coupling to make N2O, when you distort it, the one NO will just transfer over to the other ion center, but they will no longer couple. And that is now a competing reaction that is happening when you distort the core. Okay, so then we did a very deep dive into N2O yields. So we do after 5 minutes and then after 2 hours, and then we did different solvents. I don't want to, throw all the data at you, it's in the paper, but here are the most important conclusions. So first of all, if you add 2 equivalents of reductant, you get less N2O. You can see this here, this is 1 equivalent, this is 2. If you look at the 5-minute data here are the yields. You can see with 2 equivalents of reductant, you get much lower yields of N2O. If you use a coordinating solvent like acetonitrile, you get less N2O. If you look for the conditions where the dinitrosyl ion complexes are most stable, so that means you use 2 equivalent of reductant and you use acetonitrile, you look after 5 minutes, the N2O yield is actually only 5 to 10%. And so what that tells you is that in the initial reaction, making the dinitrosyl ion complex is actually the main product of the reaction. But what is weird about this is that these dinitrosyl ion complexes are not stable. You can see from 5 minutes to 2 hours, they actually decompose by making more N2O. That's also very unusual. So people have probably made 100 different dinitrosyl ion complexes. When they're mononuclear, they never make any N2O. Right? They're stable in solution in the 9 or the 10 in both redox states. So there's something special about these complexes here, that they're actually able to make more N2O over time, right? What you can see in the data. Okay, we also looked at this by EPR spectroscopy. It is known that for mononuclear FeNO2 that they are actually paramagnetic and they have an S equals 1/2 ground state. But if we take our diiron complexes and we measure the EPR, we get the same thing, right? We get a nice big signal for S equals 1/2. So we thought that when if the ion centers are coupled, you have an S equals 1/2 and you have an S equal 2, you get a spin of either 5/2 or 3/2. But we only get a 1/2. And so that means that once the dinitrosyl ion complex forms, the ions are no longer coupled, which means that the bridge, right, from the phenolate here has to be broken so that you have an isolated dinitrosyl ion complex, but then you have a non-heme ion complex here kind of hanging off next to it. And they're still connected through the ligand backbone, but there's no bridge anymore. And so the magnetic coupling now is gone, and you get that simple EPR spectrum. Okay, so DFT kind of confirms that. We did some DFT structure optimizations, and you can see from the DFT, this is the complex with the FeNO2. You can see that the distance between the ion and that phenolate here is now 4 angstroms. So the bridge is essentially broken. And if you do the same for the 10, you get pretty much a similar distance, right? You can see that. So the DFT structures confirm our conclusion from the experimental data. You can see here's the DNIC, right? The ion with 2 NOs bound, and here's the other ion which has no NO bound now, and it's just a high spin ion too. Yeah, that we figured out. Okay, and so here's a final thing that we did. So my student Corey, who did these calculations, so he looked at the calculation and he said, like, wait a minute, right? If you look at these predicted structures, this ion here from the DNIC is only bound to 2 pyridines and an amine. These are the only ligands that are bound at the DNIC, right? So he said, if that is true, then we should be able to model the DNIC by only using 2 pyridines and an amine, and we should get the same type of DNIC, right? So here's our ligand that we originally used, and now we're gonna just cut off this arm here, and this is bis-methylpyridylamine. You can actually buy this even from Sigma Aldrich. You don't have to make it yourself. So you got this ligand here, and then he prepared the corresponding FeNO7 complex. Here's a chem draw. Here's a crystal structure. And then we can now look at reducing this compound and see what happens, right? Does it give the same type of DNIC? And the answer is yes. So if you take this mononuclear complex and reduce this by half an equivalent of reductant, so that means 2 iron for 1 electron, you get this change in the IR spectrum. You don't get any N2O, and you get 2 bands, once at 1789 and 1713. And if you compare that back to our diiron complex, you can see that the frequencies are essentially the same. It's the same DNIC. We just cut off the other half from the complex. It doesn't interact with the DNIC. You get the same vibrational frequency. So that confirms directly that our DFT model is correct and that only 2 pyridines and amine is actually bound to the ion center that carries the 2 NO ligands. Right? You can do the same with the 10. I just didn't put the data in. It shows the same very close agreement. Okay, so now the final back question that you ask yourself is this guy going to make any N2O if we just wait around and see? And we tested this, and here are the data to show that. This is a control experiment. So here we take the diiron complex of methanol, we reduce it, and we wait 2 hours. We get 100% N2O, and that's the signal that you get. This is N2O in the gas headspace. All right. Then we take our mononuclear complex. We add 1 equivalent of reductant, or actually half an equivalent, or 1 electron for 2 ions, so like a semi-reduction, right? We reduce one of them to the 8, and then they make the DNIC. Do they make any N2O? And you can see here in the data, no N2O. Then we can say, okay, we can add 1 equivalent of reductant, so 1 electron per ion. We get now the Fe210, and we get 1 equivalent of ion 2. Do we get any N2O? No. So those are all the data here in black, and then this is just the background from the solvent, and you don't get any N2O, right? So that kind of is a hint what is special about our DNICs, right? If you have a DNIC and an ion 2 complex in solution, 2 free complexes, they do not make any N2O. This is what we see here, right? You get an equivalent of the DNIC, an equivalent of just free ion 2. They don't react with each other at all. So what's special about our DNIC is actually the fact that we have that non-heme ion center tethered, covalently linked to the DNIC, so it holds that other ion center always in close proximity. That's what allows then this system to go on and make more N2O over time. That's what's special about it. All right, so then we can come up with this proposed mechanism. So we start with the FeNO7 dimer. We add 1 equivalent of reductant. We make this intermediate where one of the FeNO7 is now reduced to FeNO8. It's indicated here. This intermediate, by the way, is very reactive. We have never been able to observe that. Even at -80, this never hangs out. And then with our original complex, so with the bridging carboxylate here, right, you just make N2O. If you distort the core, you open up a new reaction channel where you make some N2O, and then you start making these DNICs, either as the FeNO209 or the FeNO210. The question then is, how do we get on now from here to make more N2O? And so what we believe is what happens is actually that you can transfer the NO back to the other ion center, and that essentially you have an equilibrium like this, but the equilibrium is all the way on the top. Because if you take an IR spectrum of the mixture, you don't see any of this. So this forms in very, very, very small amount. And then once this forms, you can go back. So you go forth and back and forth and back and forth and back. And then at some point, you make a little bit of N2O. And so over time, 2 hours, then you make more and more N2O, and it kind of leaks out of this equilibrium here. So this is our proposal. We don't really have a direct proof for that, but this proposal is in agreement with our data. Right? So for example, if we use a more coordinating ligand X, or we use a coordinating solvent, then the transfer of the NO to the other side is more difficult to do, or in thermodynamic words, you shift the equilibrium over even more here, and you make less N2O. If you use the FeNO10, we know from all the studies in the literature that the FeNO29 is more reactive than the FeNO210, and the FeNO29 more easily loses NO than the FeNO210, and correspondingly from the 9, you get more N2O over time than you get from the 10. So that also kind of fits this idea. And finally, after we published our paper, just a couple of months later, there was this paper in the literature, which was pretty cool. It's the same chemistry, but you can see the ligand is completely different. So they use this pincer. This is a pincer type ligand. They have a central phosphine and then 2 files coming off, right? And they make these FeNO7 dimer complex. They add 1 equivalent of reductant, and they propose the same reaction. They get a DNIC on one side and just the non-heme ion 2 on the other side. But in this case, with this pincer ligand, this ion 2 is coordinatively completely saturated and not reactive. They can crystallize this, and they get this crystal structure. Right? So they can actually show that it's possible in a diiron complex like this, after reduction, that one NO gets transferred over to the other ion, and they have a crystal structure to show this. So although this study wasn't available at the point when we published our paper. It just came up a couple of months later. So that was very nice, you know, to see this in the literature, and it kind of confirms also our mechanistic proposal. Okay, so we can say then that we made a functional model system for these flavodiiron and reductases, and that complex produces quantitative amounts of N2O after one electron reduction, which we call the semi-reduced pathway. And then we see that if we distort now this diiron dinitrosyl core, that we get much lower N2O yields, and now we can make these dinitrosyl ion complexes. And so this could actually, or what that shows is that the reaction of making N2O is very sensitive to geometry, right? And so if you look at the enzymes, and all of these enzymes, this bridging carboxylate is always conserved, it's always there, and we can show that the bridging carboxylate actually is responsible for putting the NOs right next to each other. Because the NOs, they don't want to be next to each other, but if you bind the bridging carboxylate, it forces the NOs to be next to each other. So this is an important part in the active site of the enzyme to allow that reaction to work. If you remove the carboxylate in our model complex, we mostly make these dinitrosyl ion complexes now, and much less N2O, and this could also be a feature, right, that distinguishes enzymes, flavodiiron proteins that are more NO reducing over those that are more oxygen reducing. And then finally, I showed you we made a new type of DNIC that is actually able to make more N2O over time, which is an unusual reaction and not observed for these mononuclear dinitrosyl ion complexes. All right, so one question that kind of comes out of this is, right, what is the mechanism actually of this DNIC formation? How does that reaction really work? And this is something I would say was a more fortuitous kind of result that we got from some other studies. So my student Hai Dong, you know, here again, this is our original ligand. He wanted to make complexes where he would cut off this piece now of the ligand, where we have our bis-methylpyridine unit, but also we incorporate the bridging phenolate, but not the other arm, and then study these mononuclear complexes, and again, look at NO reduction chemistry with them. So he made a bunch of those kinds of ligands, and the ones that gave mononuclear complexes are the ones that are sterically hindered. So you put two T-butyl groups in the backbone, it forces it to be a mononuclear complex. And then here's the crystal structure of this complex. This is the FeNO7 complex, this is DNO, and you can see there's again a triflate bound to the iron center. This complex turned out to be boring, right? If you take this complex here, so this is the NO stretching frequency, the NO band by IR. You reduce with one equivalent of cobaltocene, you get the FeNO210 and no N2O at all. So this just follows the reaction that I described to you before. You reduce this to the FeNO2 come together, you get the DNIC, half an equivalent of iron 2, nothing else happens, no N2O is made, and here are the two bands. So this turned just out to be the same as the other reaction from that point of view. But here's something interesting. So then Hai went back and said, okay, what if I, take now my ligand, I go back here, and I put another pyridine here. I changed the electronics of that ligand. I removed the phenolate and put another pyridine there. And that is the so-called TPA ligand, which is one of these, famous ligands that have been used in bioinorganic chemistry forever and a day to model non-heme iron complexes since back probably the 1960s or something. So again, he made the FeNO7 complex. Here's the crystal structure. So you can see there is 3 pyridines now bound. Here's the iron NO unit, and here's again a triflate. Triflate really likes to stick to these FeNO7 complexes. Now he took that compound. Oh, and I should say this complex here is interesting also because it's close to the spin crossover point. So spin crossover means that the ion changes its spin state from high spin to low spin or low spin to high spin. So this complex has properties that are solvent dependent. So if you take an EPR spectrum of this complex in a non-coordinating solvent like dichloromethane, it's 100% high spin. So this is a high spin FeNO7 with that EPR signal around G equals 4. If you put it in a coordinating solvent like acetonitrile, you can see this signal becomes smaller, and now we see this new signal around G equals 2. This is a low spin iron 2 now with NO bound. So this complex is close to the point where iron wants to be either high spin or low spin iron 2 in the NO complex. And then you can also, so this is done at low temperature, you can also monitor this at room temperature with IR spectroscopy. So here is the NO stretching band of the high spin complex at about 1800 in dichloromethane. If you now dissolve the complex in acetonitrile, you can see there is a new band around 1700. This is the low spin complex with low spin FeNO7. So this will in a second turn out to be really important. The fact that this ion is right at the crossing point to the low spin state. So then I took this complex and reduced it by spectroelectrochemistry. So here is the NO band at 1808, and you can see nothing happens, right? There is no dinitrosyl ion complex formed. There is no high-spin FeNO8 species. These can also be stable under certain conditions, and they show NO stretching modes around here 16 to 1650 wave numbers. There is nothing there either, right? So then you probably say, well, the stuff probably just goes and makes N2O, and that's why there is no product. But if you look for N2O in the gasette space, it doesn't make any N2O either. That's strange. So I told my student, okay, why don't you go and isolate that, reaction product from the reaction mixture and, recrystallize it, prepare a sample, and we can take a look if it's just random decomposition or something else has formed. And then he came back with this NMR spectrum of the product. So you can see that this is not random decomposition or anything like this. A new product has formed. It's clearly diamagnetic, has very sharp NMR features there, but it's a new product, right? And it's diamagnetic. And then, lo and behold, we got a crystal structure, and that's what we made. It's this complex where we have a dimer now, and we have two NO minuses that bridge between these two ion centers. And these are now low spin ion 2s. Low spin ion 2d6 is diamagnetic. This is singlet NO minus, which is diamagnetic. The whole thing is dead diamagnetic, and even at room temperature, there is no paramagnetic contribution at all. Why don't we see anything by IR? Well, it turns out that when you have a singlet NO minus, the NO stretching frequency is much, much lower. And for this complex, at 1350 wave numbers. So this is very deep in the solvent window where we can't even look for any vibrations in the solution data, right? We can see this though in the solid state. So once we isolate this solid state IR, we can see this, and it's isotope sensitive, which we determined here as well. So it goes between 1350 and then 1306. So this was certainly, a surprise, and here's some Mössbauer data also that shows it's really a low spin ion 2. In cases like this, you always worry about antiferromagnetic coupling, but the Mössbauer says no, the ion itself is low spin and diamagnetic, and that is it. So that gets us then to this idea here in terms of the mechanism for Dienic formation, right? So we start with the FeNO7 complex, we reduce by one electron, we get to the FeNO8, and now we propose that these will actually dimerize to give you a structure like this, and that this is the key intermediate on the way to make these dinitrosyl ion complexes. And what happens with the TPA is when you form this structure with the TPA ligand, the ions go from high spin to low spin, and when the ions are low spin, this structure is stable. And we can isolate it, and here it is. But if the ions remain high spin, then you dissociate off this ion 2 here, and then you have your Dienic complex. All you need to do now is dissociate off the ion 2 fragment, and you have made your Dienic. So that means the Dienic formation goes through an intermediate where you have the NOs bridging between the ion centers. So my student Hai calls this spin trapping, right? He said we spin trap this intermediate. Because of the spin state change, it became stable. We could isolate it and characterize it. If the spin state doesn't change, it just goes on very quickly. You can't see this. It goes on and just makes the Dienic, right? So this is our mechanistic proposal that is based on this crystal structure that we determined. All right, I have one more part. Do we still have time or? Okay, so another interesting question is, how do we actually get, right, from this FeNO7 dimer structure, how do we get to N2O? What are the intermediates that are on this reaction pathway? And experimentally, we have no idea, right? Kurtz and his group, for the enzymes, they tried for the enzymes to trap any of these intermediates. They could not see anything. All you see is the starting complex and the end. In our model complexes, it's the same way. Even we try to run at minus 80 degrees Celsius to slow down the reaction, but all we see is the starting complex. We add an electron, we get the product. We don't see anything that's in between, right? So what are the intermediates? So the only way we were able to look at this is by computational chemistry. So in my group, we did the computations on our model system, and then there's 2 other papers where people did computations on the enzyme. But what all of these show is that the intermediates are hyponitride complexes. So once you couple the NM bond and make this N2O2 ligand. This ligand is actually N2O2 2 minus. So it takes 2 electrons from the ions and it forms a hyponitride. N2O2 2 minus is a hyponitride ligand. So we thought, okay, if we can't study this chemistry directly like this, maybe what we can do is we can take iron 2 complexes and react them with hyponitride that we preform, and then look at coordination chemistry of hyponitride with iron 2. And maybe we can learn something about this reaction. So there are some examples in the literature that are noteworthy. First one is from Richter-Addos group. So they were able to take trans hyponitride and bind it to 2 ferric hemes, and then they got this crystal structure here. So this is one example. Same idea, right? Take hyponitride and iron complex and make this. And then there's one other example from Leah's group. This is kind of a crazy compound. This is a tetramer. Each ion has 2 NOs bound, right? So this unit here, these are dinics, dinic, 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 and then there is a hyponitride that is bridging between these 2 didinic units here. This is closer to what we're interested in, but again, the dinics are not exactly electronically the same as our non-heme ion, but this is a pretty nice example that this is maybe possible, right? So we set off to see if we can do that. And the first thing we tried was going back to our good complex iron TPA. So we take iron TPA, and this is work started by Hai and then finished by Michael. And we react this with hyponitride in acetonitrile, but the sodium hyponitride does not really dissolve in acetonitrile. So this is a heterogeneous reaction. That reaction takes like 3 weeks. So you need a very good glove box, because this compound is very oxygen sensitive, and you have to stir it in the glove box in the corner somewhere in the back for 3 weeks. Then something happens, and you get a new material. You take an IR spectrum, and it has a band at 1350. You guys all know now what that is, right? 1350 band, that's our dinitrosyl compound. So what that means is, bless you. What that means is that somehow the ion binds hyponitride, it cleaves it. It cleaves hyponitride, it breaks the NM bond to make this kind of structure here. And so what we propose is that this goes through some kind of, for example, diiron intermediate here, something like this, right? And you might say, how do you know that, right? This could just be some random decomposition where just hyponitride gets cleaved and everything like kind of scrambles, and then you end up making some of this stuff, right? So in order to prove that, we did isotope scrambling experiment. So what we do now is we mix sodium hyponitride and then 50% and 50% of 15N labeled sodium hyponitride. We run the reaction. Statistically, you have 3 different products, right? You can make a product where 2 NOs are both 14N. You can have one being 14N and 15N, and you can have both being 15NO, right? And they have different NO stretching frequency predicted by, this is predicted by computation. But if it's an ordered reaction like this, where you bind one hyponitride between 2 ions and then cleave it to make this kind of structure here, right? Then you cannot get scrambling. You would only get 14N, 14N, and 15N, 15N. If it's a random decomposition, you get everything, right? So what do we get? Well, here's the IR spectrum. We get the band at 1350, and we get the band at 1325. So we get 14N, 14N, and 15N, 15N, but we do not get the mixed isotope product. And so that means, and that was kind of exciting, right? This is actually an intermolecular hyponitride cleavage reaction. It's a clean reaction where we propose that hyponitride binds between 2 ions, gets cleaved, and you get to the structure, and then this structure is very stable, and once you have this, it doesn't do anything. This is a rock, right? This is a very stable compound. Okay. Not exactly what we wanted, you might say. We wanted a hyponitride complex and not break it. So we went back and thought about, okay, you know, with the TPA is not a very strongly donating ligand. And maybe what that means is that, you know, our ion is too Lewis acidic. It will cleave hyponitride. So maybe we need to tame down Lewis acidity. And we can do that by going back to our phenolate ligands here. Phenolate is much more strongly donating, right? And so it will tamper the Lewis acidity of the ion centers. And you can directly see that by looking at NO stretching frequencies. Because if you have a more donating ligand, then the NO minus ligand will donate less, and that gives you a lower NO stretching frequency. So if you take TPA, NO stretch is about 1800. If you take this ligand here, and that's what I showed you before with the T-butyl groups, to make a monomer compound, the stretch is at 1740. So there's a 60 wave number difference between these two complexes that correlates with the Lewis acidity of the ion center. And this is also something that we've studied in broader context, and this is kind of like a side story, so I don't want to get into this too deep, but this correlation holds over the whole range of different FeNO7, high-spin FeNO7 complexes. So here we plot the NO stretching frequency against the ion NO stretching frequency, and you can see that they are directly correlated. This we measure by IR spectroscopy. This one here we measure by an exotic technique called nuclear resonance vibrational spectroscopy. And if you're coming to our workshop next week, then on Friday I will talk about how nuclear resonance vibrational spectroscopy works. But it allows us to measure ion ligand vibrations, stretching vibrations. And so this is the ion NO stretching band here, and you can see it's nicely isotope sensitive. This is done for this compound here, which has the NO ligand with the lowest NO stretching frequency. It has two phenolates bound to the ion centers. And so this actually then correlates with the Lewis acidity, which correlates with the donor strength of the co-ligand. So if you have a very strongly donating co-ligand, like here with two phenolates, then the NO- ligand donates very little electron density. So you have a high electron density in the pi star orbital, which gives you very low NO stretching frequency. And then if you go to TPA, where the ion becomes very Lewis acidic, it sucks a lot electron density from the NO-, and now you get this very high NO stretching frequency. Okay, so that just emphasizes the point, right, that we have now, by using this phenolate, really tempered the Lewis acidity of the ion center. All right, so we use this ligand here now without substitutions, and you make the lithium salt, you metallate it, and then you get a beautiful compound, which is this precursor. Here is the iron 2 complex. It has this beautiful yellow color, and it precipitates from this metallation reaction, so it's very easy to isolate in very high purity. And then, of course, we want to, here's a crystal structure that shows this dimeric structure. And then we react it with sodium hyponitrite. You get immediate color change. So you can see there is a nice shift here in the absorption band from 390 to 450. And then when we isolate the material, we take an IR spectrum. In the IR spectrum now, there is an isotope sensitive feature at about 1070 wave numbers. And that corresponds to the antisymmetric NO stretching mode of hyponitrite. So this indicates that hyponitrite is bound. And then after some work, took a little bit of time, but ultimately we got a crystal structure as well. And it turns out that we also have this kind of tetrameric structure, but it makes sense, right? Because we started from these the diiron complexes here, these dimeric complexes. So two of these complexes come together, and then they bind a central hyponitrite here. And you can better see this if we kind of draw the structure a little bit differently. Here are the two ions. This is one of the dimers. Here's the other dimer, and then the hyponitrite binds right in between those, and then that gives you this kind of structure. Okay, so finally, we took a look at the reactivity. The first reaction to try is with acids, although this, if we add acid to it, we get N2O. That's, however, it's not very surprising. You can just take sodium hyponitrite, you add acid, it will make N2O. So that is the expected reaction. What was a little bit unexpected here is that we need like 5 equivalents of acid to get up to about 100% N2O. But then again, if you think about it, we have these phenolate groups in the ligand, and the phenolate groups are basic, so they probably get protonated. And then at the end, you get only full conversion to N2O after 5 equivalents of acids are used, because there are 4 phenolates in the tetrameric structure. So this reaction is also not so interesting. The interesting reaction in terms of Flavodiiron and reductases is what happens now when you oxidize the ion center. So in this structure here, all ions are ion 2, right? 4 ion 2s. So we add 1 equivalent of ferrocenium to oxidize 1 ion to ion 3. You get some N2O. You oxidize 2 ions to ion 3, it goes completely. And so what that shows is, if you think about the reaction of the enzyme, it binds 2 NOs, it couples, it makes hyponitrite. How do you get from hyponitrite to N2O? You oxidize the ions in the process of making hyponitrite, but the ferric ions are very oxophilic. The ferric ions really like to bind an oxo bridge, and what they do then is they rip an oxo bridge out of hyponitrite, and that leaves you with N2O that will take off. So we proposed that the oxophilicity of IM3 is an important driving force for the second part of the reaction, which is the decomposition of hyponitrite to actually make N2O. Okay, so we can summarize this part. So I showed you that with this BMP phenolate ligand, we're able to make a stable hyponitrite complex. And we see it has this tetrameric structure. Oh, I forgot to mention this. If you look at the structural parameters of the bound hyponitrite, it just looks like sodium hyponitrite. So the hyponitrite is not activated in the complex of iron. Structurally, it looks the same as if you just have sodium hyponitrite. And then if you add acid, you make some N2O. That's the expected reaction. More interestingly is that the hyponitrite ligand is sensitive to the Lewis acidity of the iron centers. If you have iron 2s that are Lewis acidic, like in the iron TPA, you cleave hyponitrite and make this diamond core structure. But if you go to iron 3, and iron 3 is very oxophilic, it likes a bridging oxo or hydroxyl ligand, it will rip an oxo ligand out of hyponitrite and it will cleanly make N2O. So we propose that the oxophilicity of iron 3 is an important part of the reaction that ultimately leads to N2O formation in the enzyme. Okay, and that's the end. So here's a picture of my current group. Here are some of the students I highlighted on the way that worked on this project. Dr. Corey White, Dr. Haidong, Dr. Amy Spielman did that initial experiment with that crystal structure of this core-distorted complex. These are all former students. Current student Michael Lengel, and he is right there. And thank Carsten Krebs for collaboration. He did the Mössbauer spectroscopy. We have a pretty big group of people doing bioinorganic chemistry at Michigan. So I thank those guys for lots of insightful, discussions. I like to thank the funding agencies. They still exist, but who knows how much longer. So, but for now, we have to thank them. And I thank you all for your attention. All right, and time for questions. Antonio will manage.
So if you're not comfortable to ask in English, you can ask in Portuguese and we can help and
Cheers.
you can also try.
Hi, Nicolai. Thank you for your nice presentation.
Thank you.
I have some curiosity. When you say that even at -80 degrees, you do not see the intermediate, you cannot isolate or you cannot
So that,
observe
yeah.
that. But have you tried doing a freeze quench at low temperature?
Oh, okay. So you think like reducing and freeze quenching after like a millisecond or something.
Milliseconds,
Yeah, we have not tried that experiment. I don't have a freeze quench apparatus in my lab, so we weren't able to do that. So we tried to just, lower the temperature to see if it would stabilize, but that's not the case.
You could run the reaction to low temperature and freeze quench at a shorter time, like milliseconds, like 30 milliseconds or something like
Well,
that.
yeah, that's one. I had another idea to do that, which was a photoreductant,
Yeah, that's also,
right? So
yeah.
we have a photoreductant. When you flash it with a laser, in the excited state, it will reduce the iron complex. So you can mix them in the dark and they're stable. You could put them in a spectroscopy cell, you flash with the laser, then you reduce it to the 7-8, and then you can use much faster spectroscopy. So that's kind of what I was thinking about doing maybe at Argonne National Lab with X-ray spectroscopy. But yeah, so this certainly also
Also,
a way we could try, right?
It could be tried. We have the flash photons also for that. So.
Yeah, for milliseconds. It's good. We cannot do milliseconds.
And the other question that I was wondering, you have plot the stretching frequency for NO
Yeah.
versus the stretching frequencies for metal, nitrogen,
Yeah.
for metal
This
NO,
guy
right?
here.
Yeah.
Yeah.
You got to this iron NO stretch frequency from nuclear resonance vibrational, but could you see? Could you not see, but by infrared as well, or by Raman?
So by infrared spectroscopy, we were not really able to see, a nice band. There's some very, very small, tiny features, but then you always worry about if there's some decomp going on. By Raman, we could not see anything. And so it turns out then by nuclear resonance vibrational spectroscopy, these are very easy to measure. They have a lot of ion motion, so they give very big signal, right? So that was then the technique we used for all of these compounds that are shown in this table.
Just curious because we could see that
graph.
distortion for ruthenium NO in our systems using
Yeah, we
Raman
cannot do nerves. Yeah. But for that one, you can probably do non-resonance Raman. So we had some success in some other study measuring the ruthenium NO stretching frequency just by Raman with near-infrared excitation. So that's the way to go there. We did not try this on these compounds. So then we took them to the beamline and did the nerves experiment, which then worked out really well. Thank you.
Questions should be not ashamed. Can ask. I can push him.
Whoever asks a question, here we have some some cake. Whoever asks a question can come and get some cake.
Yeah, cake and pão de queijo. So, I mean, for me, like I'll ask a question and curiosity also. Like for me, it's interesting how you saw that like there's the angle of NO binding the iron is like making your reaction occurs in one direction or another one is like pretty interesting. I think it's like, is this only be seen by iron? Have someone else seen this by other metals or maybe
So for
copper
these-
or I don't know? It's like, is this only for this kind of system or these enzymes?
So I think, if you look at different metalloenzymes that do these kind of nitride or NO reduction, and you look at the products versus the metal, there's some really interesting correlations, right? So for example, if you want to make NO from nitride, you should use copper because copper stops at NO and it will not go further. If you use a heme, you'll go to ammonia or some, intermediate species. If you want to take NO and make N2O, these diiron systems have that selectivity. They go to N2O, they don't go any further. And you could, of course, reduce NO also then to hydroxylamine and ammonia. They don't do that either. So this seems to be a chemistry that you can do with the iron systems. You can also do it with a heme and a non-heme ion. There is this NO reductase with heme and non-heme ion that also goes on to make N2O. And I think you can also do a heme and a copper in that enzyme. And I think that makes some N2O as well. But these non-heme diiron systems are very good at that, right? This reaction, when you add an equivalent of reductant, we did some. So we don't have freeze quench, but we did some stopped flow IR experiments. It can at least show that the reaction is done in the dead time of the instrument. So 150 milliseconds, the reaction is done. So this is a very good reaction to make N2O. But it was surprising, that just the angle, right? You change the angle and now you're changing the product so that there is this really, very strong requirement for geometry in order to make this to work. And I think that that's one important difference between reducing NO and reducing oxygen, right? When you reduce oxygen, you take one molecule of O2, it bridges between the ions. I think you can have different structures, doesn't really matter, right? It will make a peroxide, break the bond, and go on. But for NO reduction, you really have to have them right next to each other. As soon as you distort from there, the reaction breaks down. So we did not expect that.
Thank you. But thinking about your experiments, I think I never did this kind of experiments. You were seeing like the N2O that's being released by your reaction that's made at -80, something like that.
Yeah,
like
the
70.
reaction still works at -80.
So do you do like stopped flow and then you freeze it and then you increase the temperature to -80 and see the top of your cell? Like how is I mean, do you have like a draw or something of how is this experiment done?
So
like,
I mean, the gas phase IR experiment is actually very primitive, right? So all you do is you have a closed reaction vessel. You let the reaction run for a certain amount of time, and you can do room temperature or low temperature, doesn't matter. And then you connect it to a gas IR cell that's under vacuum, and you suck the gas headspace into that cell. And then you have to make a calibration curve, of course, right? You have to be very good with a calibration curve. You measure the IR signal for N2O, you integrate that signal against the calibration curve, you can determine the yield from the gas headspace. And so that's how we did it at, -80 degrees Celsius. So we just, made a cold and acetone dry ice at the reductant, wait a certain amount of time, and you get that. And then there you go. The other possibility is to do a solution IR spectrum, because you can see N2O product, you can see actually in IR, right? So You run the reaction -80, you take some solution out, put it in a cell, all cold, measure NIR, and then you have a giant N2O peak. So you can also show like this that the reaction does actually proceed at -80 even. It does not stop after reducing the NO. But we have some hope that compound that Philippe made with the carboxylates that might be able to trap the NO before N2O is made.
Someone else? Oh, good.
First of all, welcome to Brazil.
Thank you.
Sir, the presentation was nice and it was totally structural inorganic chemistry. Sir, my question is about, as per your expertise, it is biological side.
Okay.
Okay. So my question is, sir, what are the impact of nitric oxide inside our human body with the enzymes?
So nitric oxide is made in our body, right, for the signaling and then also for immune defense.
Yes, sir.
And for immune defense, it's made in like macrophages or other immune cells.
Yes, sir.
And then they release NO and superoxide and other toxic chemicals, and they try to kill, pathogens that infect our bodies, right? So it has a very important role in immune defense. Yes. And so here, some pathogens now can detoxify NO, right? By reduce it to N2O, it's no longer toxic to them, right? So this is essentially a defense mechanism against NO. So these bacteria, they have transcription factors. They can sense NO.
Okay,
To expose them to NO, they can sense it. And then once they sense it, they go and they make these flavodiiron proteins as scavengers. So they pick up NO and reduce it. So it's no longer toxic to these microbes, right? So this is an interesting link to infectious disease.
Yes.
Not all pathogens have them, but there's certain type of pathogens that have these kinds of enzymes available to them.
Yes, sir. Sir, we know the excess production is toxic for body, the nitric oxide. So is there any natural way to overcome the production of nitric oxide?
I mean, the way you could do that is, if you're looking at the NO produced in our body, would be by inhibiting the enzymes, right? So the enzymes that make NO are called nitric oxide synthetases.
Okay, sir.
They take L-arginine and oxidize it to citrulline, and they release NO, so two-step oxidation. And so these enzymes are found then in macrophages, in your endothelial cells, in your brain.
Yes, sir.
And people are developing selective inhibitors for these enzymes as therapeutics, as drugs.
Okay.
And some of them are already sold now, because they can also then help with other health problems, right? Because if you're making too much NO in your body, at some point it becomes toxic, right?
Oh, yes, sir.
And so that is one of the problems also with chronic infections. You have a chronic infection in your knee, right? So it stays infected for a long time. The whole time your body makes NO trying to kill these pathogens, but it can't.
Yes.
Over time, the NO will start damaging your body, right? It will damage the cartilage. It can also, if you have infection of an organ, it can cause organ failure,
Yes, sir.
right? And so that's why there is an interest also in diabetes.
Yes, sir.
There's overproduction. So there's an interest to make drugs that inhibit NO production. And it's very difficult, but there are some now available.
Yes, sir. Thank you so much, sir.
Yeah, of course.
Big. So, oh,
There is
Nicholas, nice. Good, good.
Oh, come on.
Not at all. So thank you for your presentation, and actually not for the field. So there's a huge probability that I lost something in your presentation, but actually, I need to do to make a question a bit stupid, but
It's okay.
no, actually it is. But in the end, mainly for the first part, are you trying to understand what is the active sites of the enzymes, like synthesizing the complex and mimetizing the active sites, try to understand? Or is just similar complex and you are studying the annual
Feedback.
reactions?
So we don't, well, it has two aspects to it, right? So we start, of course, with we're almost there. We need to know what the structure is in the enzyme, first of all, right? So otherwise we don't know what the catalyst, we go back. Nice light show. So first of all, we need to know this, right? We need to know what's the structure, what's the catalyst that does the reaction so we can model it, right? But our goal is to understand the mechanism of the reaction. How does it work? And then the other question is, what are the factors that contribute to allow that reaction to happen, right? So for example, what's very important here is the redox potential of the diiron core. I didn't talk about that today, but we also did some studies where we started varying the redox potential and it affects the reaction, right? And then, yeah, it's not shown in this graphic here, but there is a second coordination sphere group, a tyrosine, that sits on top of one of the iron centers and it's proposed to make a hydrogen bond with one of the NOs, right? That's another aspect. So how does that affect reactivity? How does that affect NO activation? And then if we go here, that's the mechanistic overview. We're interested to see, right? How does the reaction work? And can that reaction work this way or this way or this way? And what I talked about today is the reaction can go this way with this one-electron reduction, but if you tune the iron center just right, you can actually go this way, which we discovered but I didn't talk about today. Otherwise, we have here a 2-hour presentation, and nobody wants that, right? So I picked that topic. But we also looked at this point is a crossing point, right? At this point, the road diverges, and you can actually, by changing the property of the diiron core, you can determine which reaction is going to go, right? So it really gives you that fundamental understanding of, geometric electronic structure of iron NO complexes and the requirements in order to ultimately make N2O from this reaction. And then the other interesting aspect from a fundamental science point of view is, are there other reactions that can happen in that space? Right? And we discovered one other reaction is when you distort the core, you can make the nitrosyl iron complexes. That's not observed in nature, but it's another reaction. It can give you some idea to think about, why is, if we go back here, right, you can ask the question, why is the structure this way, right? And we can answer some of these questions. We can say, oh, this bridging, where is it? The bridging carboxylate. If you take that out, the reaction will no longer go, right? Because you can distort the core, then you don't make much N2O anymore. So, right, we can see structural requirements here, electronic structural requirements. What are the ingredients in order to take NO and make N2O with a non-heme diiron core? What are the different reactions that can happen, right? So it's very fundamental science. It's really trying to understand the fundamentals of this chemistry.
No, perfect. Actually, it's similar what I think about, because I remember a little bit about the cytochrome C oxidase, for example. And I remember to see the story like, it's iron, it's the active site, but it has copper. So oxygen, it's the dioxygen to be react. It's binding to the copper, or it's just electron influence. And after discover, I think the tyrosine, something like that, the influence of the structure itself.
Yeah.
So my question would be like, what do you think your models, how far they are from what could be the real active site, if you can know that or not? And just
so the models are never exactly the same as the active site, obviously, right? For one thing, we don't use the amino acids, and they're not embedded in the protein matrix, so we don't have the same structure. But we can use our models to study the same chemistry. And then the other aspect is, this is what the wild type enzyme looks like. So how, now you want to change this structure. That is not easy, right? Yeah. You can do site-directed mutagenesis. You can take one amino acid and turn it into something else. But oftentimes, the whole structure then around it will rearrange. And you can't really exactly change one thing about this. You say, oh, I want to change redox potential. I want to add a hydrogen bond or take it away. As soon as you do any mutagenesis, the structure changes much more than people appreciate. But in the model complexes, we can do exactly that. We can probe, right? We can take we'll go back to the ligand. Wait, no, where's the draw? Oh no, it comes later. Wrong way. So we can take this complex now, and we can make changes to this complex without changing the overall structure. So for example, we can change donor strength here, putting like a phenolate there, we can influence the redox potential without influencing the structure much. Right? We can put like hydrogen bond donor in the second coordination sphere to see what if I form a hydrogen bond with NO, right? And we can see how that affects things. We can also take a protein out, put a carboxylate there, all different things we can do, and we have a great level of control over the overall structure and property of the complex in that way. So with the model complexes, we can much easier ask these really fundamental questions, right? How is the electronic structure or geometric structure linked to reactivity? And we can ask and answer in a very clear way. But with the enzyme, it's often very difficult, right? I mean, if you I have another project we do on biocatalysis anyway. So we do some mutagenesis and we get very strange reactivity. Then we run some molecular dynamics. We see that the structure of the active site is completely different. As soon as you take one element out, you have some hydrogen bonding network. All the groups, part of the hydrogen bonding network, will rearrange to some other structure. And then the whole structure can collapse or expand in a very unexpected ways. So using mutagenesis to really answer mechanistic questions is much more difficult than a biochemist tell you it is. So for this fundamental correlation, the model complexes give you a much better level of control over the properties.
Yeah. No, I understand that. I mean, even if you're saying like just putting some things I can imagine that's very hard to synthesize. It was just curiosity, but yeah, thank you.
But yeah, you always keep in mind it's not exactly the same thing. So we are very interested, in the data that the biochemists have collected for the enzyme. You look at all the data, and that's something you take into consideration, right? Of course.
Someone else? I'll ask one more.
All
so
right.
do you have you were talking about redox potential, like are people able to realize what's exactly the redox potential of this iron in the enzyme?
Yeah,
By
there's some
electrochemistry?
there is some data available for that, and we can compare that to the model complexes as well. So if you
do this by electrochemistry, or you use like oxidants or reductants and see the potential?
I think
I don't know how it's.
I don't remember if they immobilized the enzyme or did it with a redox mediator, one or the other, but it's done with electrochemistry, one of those ways. And it turns out for this, as long as you keep the NOs, next to each other, you don't disturb that. The redox potential is what can send it one way or the other. So the complex that I talked about today, the ions have relative positive redox potentials. So with this complex, you can't really make an iron-free, because that redox potential is like +1 volt versus ferrocenium. So that core only provides one electron for NO reduction. That means you have to add another electron, right? And then it will go. If you now change the ligand environment to push the redox potential more negative, then you can open up that other reaction channel. Right now, the complexes that we had, one has 2 positive and one has 2 negative redox potentials, and the enzyme is in the middle. But the complex that Felipe was working on is the magic one. It's right there where the enzyme is. But it has some other problems, which is why we're still working on it. So that's not ready for prime time yet, because iron really likes carboxylate, okay? Iron loves carboxylate. When you have carboxylates there, they're unprotected. Iron likes to bridge. And so if you have free carboxylates hanging out there, then these diiron complexes will form larger structures by bridging through these carboxylates, which of course you don't want if you want to study the iron complexes. So we have to find a way to shut down this oligomerization process, something that we initially didn't realize, but now we know. So we have some work going on now to make, the nice dimers, and then we can study, chemistry with them. And that was one of the reasons why in Felipe's work, the N2O yield was low, because the structures are different from what we thought they were. You have to be very careful with those kinds of things, right? But yeah, redox potential is an absolute important feature for that reaction.
One else? So thanks again, Nicolai, for an amazing talk, and answering the questions. So now we can have like a pão de queijo and cake.
Thank you. Now we can have the cake.